Good morning to all. Myself Janarthanan, Professor, Department of Pharmaceutical Chemistry, Prime College of Pharmacy, Palakkad. Today I am going to take on the topic of artificial enzyme. Enzymes are biological catalysts. It can be defined as a substance that speed up the rate of chemical reaction without involving that. So enzymes can catalyze all the chemical reactions taking place in the cells of the body. They play a vital role on the functions of digestion and metabolism. So artificial enzymes can be defined as synthetic organic molecules which mimic the active sites of natural enzymes. Artificial enzymes need to bind molecules made based on host molecules such as cyclodextrin, crown ethers. It has molecular weight less than 200 daltons and it is stable at high temperature, and they are also known as enzymes or enzyme mimics. Enzymes are capable of cutting and joining strands of RNA in test tube and used for treatment of cancer and viral infections. So for ideal characteristics of artificial enzyme, it should have hydrogen bonding or electrostatic binding sites. It should have catalytic group attached to model. It should have a rigid model, water soluble and active at physiological conditions, reversible and non-covalent binding. So design approach for artificial enzymes, there are two types of approaches. The first one will be chemical approaches and second will be biological approaches. In chemical approaches, we have cyclodextrin, cyclophane, calixarein and crown ethers as enzyme mimics of chemical approaches. And biological approaches, there are two types: direct evolution method for artificial enzyme production, de novo computational enzyme designing. So indirect evolution method for artificial enzyme production, it is a molecular biology method to modify a biocatalyst via Darwinian evolution. It has following steps: a gene mutagenesis, insertion into host, transformed colony. So examples of this direct evolution method for artificial enzyme production is sitagliptin. It's used for treatment of type 2 diabetes as well as blood pressure. The second will be de novo computational enzyme designing. It is an automated identification of amino acid sequence performed that will fold into specified three-dimensional structures. It is a method emerged as a promising tool for engineering enzymes. The following tools are used: path-pred RDM pattern, bond electron matrix, reaction smarts in retro, path 2.0. So the applications, if you see that pharmaceutical application used for development of drug and chemicals. In medicine, it's used as supplements for patients deficient in certain enzymes. And in genetic engineering, it is used to create a genetically modified organisms or to help genealogy research. So thank you all.